Proteins are large complex molecules made of many polypeptids which are chains of amino acids. They play many important roles in the body. For example, they act as enzymes to speed up chemical reactions, they build and maintain cell structures such as collagen, they act as chemical messengers, and are key components of the blood. Amino acids are the monomers from which polypeptids are made. There are 20 different types of amino acids found in all living organisms. Each amino acid has a similar structure with a central carbon atom, an amine group, a hydrogen atom, a carboxyl group, and a variable side group that makes each amino acid unique. Polypeptids are formed through a condensation reaction of many amino acids, which creates peptid bonds, a type of covalent bond. A dipeptide is formed via a condensation reaction between two amino acids, also creating a peptid bond. Proteins have 4 levels of structure: the primary, secondary, tertiary, and quaternary structures. The primary structure is the sequence of amino acids in the polypeptid chain joined by peptid bonds. The primary structure of a protein determines its tertiary structure. The secondary structure is the folding of the polypeptid chain into an alpha helix or a beta pleated sheet, due to hydrogen bonds forming between the groups in the peptid bonds. Hydrogen bonds are weak attractions between a slightly positive hydrogen atom and a slightly negative atom, usually oxygen or nitrogen. In this case, the partially negative atom is the oxygen in the carbonyl group. The partially negative oxygen in the carbonyl group and the partially positive hydrogen, the one bonded to the nitrogen, are attracted to each other and form hydrogen bonds. Hydrogen bonds help to maintain the protein's shape when they are folded into alpha helixes or beta pleated sheets. The tertiary structure is the folding of the entire polypeptid chain into a specific 3-dimensional shape. This folding occurs due to a combination of hydrogen bonds, ionic bonds, and disulfide bridges forming between the amino acid side groups. Disulfide bridges are strong covalent bonds formed between the sulfur atoms of 2 cysteine amino acids, creating a bridge between different protein parts. This bridge provides extra stability to the protein's final shape, ensuring it stays intact under tough conditions. Ionic bonds are attractions between parts of the protein with opposite charges. They help to keep the protein folded correctly by holding different parts of the protein together. The quaternary structure is formed when 2 or more polypeptid chains bind together to create a larger functional protein. Each polypeptid within the quaternary structure still maintains its own folded tertiary structure. However, not all proteins have a quaternary structure. Proteins made from a single polypeptid chain have their tertiary structure as their final 3D shape. On the other hand, proteins made from multiple polypeptid chains have a quaternary structure as their final 3D shape. A protein's final shape determines its function. Once folded into their 3D structure, these proteins can perform their specific roles in the body. Proteins that are fully folded and can carry out their specific biological role are called functional proteins. Examples of functional proteins include enzymes, transport proteins, structural proteins, antibodies, and hormones. There are 2 factors that affect protein structure: the primary structure and pH. When the primary structure of a protein changes, the sequence of amino acids is altered. This can disrupt the protein's folding, breaking the bonds that hold its 3D shape. As a result, the protein's tertiary structure changes and becomes non-functional, impacting its ability to perform its biological role. The primary structure of a protein can be altered by gene mutations or mistakes in mRNA translation during protein synthesis. Changes in pH can break the ionic and hydrogen bonds in a protein, altering its tertiary structure and making it non-functional. However, disulfide bridges aren't affected by pH. This is because they're strong covalent bonds, which in comparison to ionic and hydrogen bonds, are stronger and much harder to break. Proteins can have non-protein parts called prosthetic groups, and when they do, they are known as conjugated proteins. Hemoglobin is an example of a conjugated protein found in red blood cells. It has a quaternary structure made up of 4 polypeptid chains called globins, each with a heme group. Each heme group contains iron and can bind to 1 oxygen molecule at a time, allowing a single hemoglobin molecule to carry 4 oxygen molecules. The heme groups are the non-protein components in hemoglobin that make it a conjugated protein. Hemoglobin is also an example of a functional protein as it performs a specific role, transporting oxygen. We use the Biuret test to check for proteins. Here's how to do it. Put a food sample into a test tube. Add an equal volume of sodium hydroxide solution to the test tube, which creates the alkaline conditions needed for the test. Then, add a few drops of copper sulfate solution, and gently mix. The combination of sodium hydroxide and copper sulfate is known as the Biuret reagent. In the exam, rather than saying add sodium hydroxide and copper sulfate, you can simply say add Biuret reagent. If proteins are present, the solution will change from blue to purple. If it stays blue, no proteins are present.